So today I'm not here to sell you an expression system, a media feed or supplement, but more to give you details about how you can design experiment to improve your media and your feeds. So today I will first. Okay. So first I would like to introduce my company and where I am working. So I'm working at Merck Serono. It is the largest division of Merck KGaA, the German company. I am based in Switzerland. I'm working in Vevey, and in Vevey we are mainly doing production, but there is also a team of about 60 people that are working in process development, and where it is where I am working. Today I will present you, I would like to give you an idea of how you can design experiments to improve your media in your feed using mainly high throughput cell culture method. So I will be mainly talking about 96 deepwell plates, and I will illustrate my presentation with two case studies. The first one is about high throughput media blending. It is a work we have done last year, and that will be published very soon in MABS. In the second case study, I will be more touching about the product quality optimization that we can also maybe handle with high throughput cell culture system. So here first it is an overview of our concept of development. It is what we call an integrated development approach. As many of you, our goal is to obtain, to deliver high performance cell line to manufacturing, high performance processes, and deliver the right quality. So for this, at the different step of the process development, from cell line generation, evaluation, through medium development, feed development, process development, and also process validation using QBD, quality by design, we have implemented a set of tools of culture method to improve our development, to get the maximum knowledge about our processes. So you can see here some of the tools that we have implemented. I was talking before earlier of 96 deepwell plate that are automated with robotic platform. We have also heard today about the Amber system from TAP, and also the check tubes that are a really good and high throughput cell culture model, small scale model, and also, of course, the benchtop bioreactor. In top of that, during the last year, we have really improved our way of we developed our processes with improved design of experiment using media blending, feed blending, DOE, but also we try to make predictive the smaller scale, like 96 deepwell plate, of the manufacturing performance. So we are implementing also computational fluid dynamics to really fully understand what is going on from the really small scale, smaller scale to the manufacturing. Here it's an example of how we plan to select our cell line. So recently you may have read some paper about the predictability of static system compared to fed batch culture, or even of batch culture to fed batch culture. So here the goal is to implement early as possible a fed batch screening in 96 deepwell plate. So just after transfection cloning, we adapt the cell in 96 deepwell plate to run a fed batch culture very quickly, and to go down from 500 clones to 12 very quickly. And then it is a standard workflow. We go into shake tube, microscale bioreactor, and bench scale bioreactor to select our final cell line. Now, the first case study So as I said, it is a really new data results that will be published in MABS really soon. I don't know yet the date, but it will be available soon. Here we'll be discussing about media blending, but in high throughput mode. You certainly know the media blending are taking 5-6 commercial media, blending them together to see what is the best combination. Here we are mainly focusing on the development of our proprietary medium. So we are targeting to fully design our medium. So here is the workflow. We are starting from our 47 components that we have in our proprietary medium, the first generation, I will call it like that. And we will make 15 formulation. With this 15 formulation, we will blend them in a deep well plate to evaluate them in a fed batch culture. And we are evaluating about 400 culture in parallel. The most important thing here is the design of the 16 formulation, media formulation. First, we are varying in this 16 formulation 43 components out of the 47 of our medium. Each component is set at 3 levels, a low level, a mid level, and a high level. This is based on literature, on previous experiences, and then we have to design the 16 formulation. This has been done randomly, but to avoid correlation between the component when you are evaluating your 400 media component, we have designed about 80 set of 16 media formulation. And with this different set of media formulations, we have in silico look at the correlation between the component in our 400 media final blends. And doing this, we have selected the set of 16 media formulation that reduce at maximum the correlation between the components. It was important for us because we wanted to know what are the key components in our process optimization. In terms of cell culture, here is the protocol. So we did 3 passages prior to the fed batch inoculation. So it was made to adapt the cell, or mainly to prevent carryover effect before to inoculate the fed batch process. This was done in 96 dipole plate, as I already said, and each culture was diluting after each passage at a targeting cell density. This was done to guarantee that the experiment is made as you will do it at larger scale. And for the fed batch process, on day 2, 4, 7, and 10, we have added a reference proprietary feed. This was to obtain a medium that is really adapted to our feed system that we have already in place. For performance assessment, we did cell counting, cell viability using the guava, and titer quantification with the octet FortiView. Here you have an overview of the result that we obtain. As you can see, the range of result is really big, from almost no growth to almost 25 million cells per ml. And for titer, it was the same, from very low titer to up to 3 grams, 3.5 grams per liter. Here you have 2 controls. In blue, it was our 3rd generation proprietary medium. And the second control is one of the 16 formulation. It is a formulation where all the components are at the mid-level. To analyze the data, we use three ways of analysis. The first way is quite simple. You take your result, you rank regarding titer, IVC, viability, and you identify the best condition to go further. So you just select your best result. In the second approach, something I didn't mention here, we blend the 16 formulation together using a mixture design. So we were blending 2 medium together in a 1:1 proportion and 1:2 proportion. So it's a kind of design experiment. So with tools such as the Design Expert software, we can really analyze our data and identify the key medium formulation out of the 16 to identify the key media formulation, but also to predict, to make prediction, to see if I need 1, 2, 3 media formulation and what proportion of each one. And the third way of analysis was doing multivariate analysis. So here, the goal was to identify the key components that drive our performance down or up. So here, the output of this was a list of components that could be interesting to further analyze to improve your process. So here, it's an overview of the result that you can obtain with the design of experiment approach. So here, first, you have the model obtained for the titer with the observed value and the predicted value. As you can see, the prediction is quite good with the R2 of 0.9 almost. With that, we can do prediction. So here we have predict the 5 top prediction to maximize the titer. You can see that some formulation was not really useful to drive our performance up, our titer up. So here, formulation 2, 3, 5, 10, 13, and 16 was quite important in our experiment. The average is just the proportion of each formulation for the average of the 5 first prediction. So the DOE analysis allows us really to identify the key formulation from the 16 basal medium. It allows us to design very quickly a new media formulation based on the prediction, but it doesn't allow you to fully understand what is behind this, which component is really important in your process. So that's why we did the multivariate analysis. So with the PLS model, here it's for titer at day 14. We rank the component by influence. So here you have the positive component in the model, and here you have the negative component in the model. So what we did here is we selected the most important component in a range. So you have ferric ammonium citrate on one end and serine on the other end. But as I said before, there is, by design, some correlation between components because we have a very low number of conditions tested compared to the high number of factors tested, 43. So here, I look at the correlation between components, and it appears that some components in yellow were very correlated with some components in orange. So, highlighting these 13 components, we have a list of components that are certainly that require certainly a further optimization to obtain the best performance. Here's just an example for ferric ammonium citrate. You can see here that at the lower concentration you obtain always very low titer, whereas at the maximum density concentration, you obtain quite high titer. And you can see also 2 thresholds here and here. You have a broad range of performance. Your process is not really robust in this range of concentration for ferric ammonium citrate. So this is the kind of information that you can get out of this kind of experiment. We have performed scale-up experiment and confirmation. And so first, we select with the empirical approach, the ranking approach. We select 8 media, and also 1 from the prediction with the design expert approach. From this, 1 medium shows really interesting feature, so we go up to bioreactor, and we even evaluated it on different cell lines. So 3 cell lines were evaluated, and we obtained improvement from 30 to 60% depending on the cell line. So it's that you can see here. We obtained for 1 cell line, the one in red here, even 5.2 grams per liter in bioreactor. So it was quite nice for us. In conclusion, media blending is a really powerful tool. It's useful when you can do design of experiment, mainly when you have solubility issue for some component. Sometimes you can solubilize in a stock solution tyrosine. It's quite difficult. So media blending allow you to design completely your medium, taking in account all the components of your medium. We can imagine the same approach for the feed. It would be even more interesting regarding the solubility issue. And we can even imagine a combination of both blending. If you took the result from this experiment, 5 formulation was really important to drive our performance up. If you took the result from the feed blending experiment, maybe also 5 formulation will be interesting. You take these 10 formulation, we mix them together, and you maybe obtain something really, really interesting. The second case study is about product quality, because here I only talk about process performance. Here, I wanted to show you what we can do with high throughput cell culture methods such as 96 deepwell plate for product quality optimization. So here the context is we have a process A. We wanted to go to a process B, more performant, but we also wanted to keep the product quality constant because we already started the clinical trials. What are the challenges with high throughput cell culture method and quality analysis? Usually, you obtain really low product in 96 deepwell plate, amount of product, sorry. So it's about 300 microgram, sometimes 1.5 milligrams, but it's quite low. So if you want to do quality analysis, you usually need a huge amount of product. So here we are using some kind of fingerprinting method to assess the product quality. So we do the charge profile with the ICE280, glycan analysis using capillary gel electrophoresis, and also product integrity using Sekech PLC or caliper. What we did is also with about 400 samples of about 400 microliter, we achieved to perform all this analysis in one experiment. It takes about 2 weeks and a half for our analytical lab to perform all the analysis, and about even 9,000 results were generated. So I will just show you the design of the experiment. Here, it was not just a media blending, but a combination of media blending and DOE experiment. So here, we had 5 basal media. At the beginning, we mixed them together to obtain 11 media. And on top of that, we add a DOE testing 17 factors that were added at day 5 on top of our standard feed. I already mentioned the analytics that we performed. It was only performed at day 14. I will only show you the result for the charge profile and for the cluster 4. So the peak 4, it's kind of a basic isoform of the molecule. You have here all the data obtained over the 400 condition, so it's a broad range of results. And here first you have the control, it's replicates of the basal medium, of the basal media. You can see very small variation. But here, when you have the different media and on top of it the 32 conditions that test the 17 factors, you can see that you can obtain a larger variation. And if you are targeting to be in this range here in green, you can see that some conditions enter in the target product profile. So for each CQA, we look at the distribution of the results. We also did models. So here, for example, also for the cluster 4, you can see which component drive up or down the product quality. So here it was interesting to see that adding copper in our feed, we can improve or at least came back into our target product profile. It was the same with permine, but with an ACL, it was not right. So it means that we had to decrease the osmolarity of our feed. We have seen also some effect of the medium. For example, basal medium 4 was really interesting with a smaller, providing a lower number of cluster 4 isoform. So we did this for each CQAs. It was a huge analysis, but in the end we managed to fix with the model the level of 4 factors. A number of factors were eliminated, and we selected also 4 factors and 2 media for further optimization. This was done at larger scale using the Ember system. I don't have data here, but after optimization, we obtained a process that was successful and that can be transferred in manufacturing with the correct product quality. So in conclusion, what I would like to say is that now high throughput tools are really here to perform powerful experiment, not only for cell culture but also for product quality. And what is important for me is that designing the right experiment, you can get really the maximum of this kind of tools. For the moment, at Merck Serono, we mainly use this tool for media design, but in the near future, we will do it also for feed and process. So we could even have more interesting results doing this. I would like to thank the people that work with me to obtain this data from the Upstream group and from the analytical group. And thank you for your attention. I will be happy to take your question.